Hi everyone, Dr. Hinkey here with today's video lecture of Chapter 13, The Control of Microbial Growth. So we've looked at all the conditions that microbes need or that we can give them for their happy place so they grow at their optimal rate. And now we're going to look at how we can control that growth. So our objectives, we're going to, for this first portion, we're going to define some terms that are associated with control of growth and with the spread of pathogens, and look at different safety levels. A lot of this unit is vocabulary, and a lot of the vocabulary, if you look at it, sterilization, aseptic technique, asepsis, sepsis, disinfectant, antiseptics, disinfection. They're terms you are familiar with. But now in microbiology, we are going from the common day-to-day use of them, how we would just colloquially use them almost interchangeably, to specific meanings related to controlling microbial growth. Sometimes this makes it more challenging because you already have some idea of what the words, of what you think the words mean in your head, of the common use. Now we're getting to a precise use. So you might have to unlearn what you think some of these words mean in order to learn the accurate microbial growth definitions. So the reason we want to control microorganisms is to prevent the spread of disease. So we have to particularly pay attention to items that are frequently used and frequently come into contact with humans as a way to get those pathogens to a portal of entry to where they can get in and cause infections. So one of the most common ways that we pass infections is through fomites. A fomite is an inanimate item that can harbor microbes. So they can be there. We don't have to come in direct contact with someone who has an infection. They can leave microbes on something they have touched or used, and then later those are passed on by someone else who uses that same object. So fomites mean we don't have to come directly into contact with the actual bacteria. So these are an indirect method of transmission. So, and here's some common examples. Keys that you touch and pass, leave around, other people have touched. Towels, door handles are a big one. So anything that you touch and pass around. That's why there's a lot of concern. A lot of places are going to no contact or touchless payment, because money is a big thing that everybody touches and passes through lots and lots of people, lots of hands. So we have two different factors that influence the level of cleanliness when we're looking at cleaning these fomites, these different indirect transmission objects that microbes may survive or grow on. So we have to think of, how will these items be used? Items that come directly into contact with internal organs. So if it will be inserted into the body or come into contact with, say, mucous membranes, or internal, like catheters, requires a much higher level of cleanliness. And then we need to consider what pathogens could come into contact with this fomite. So are there potentially spore formers present? Is this somewhere where a bacillus that forms spores, where something that's spore forming is usually found? If it is, we know they're more resistant. They're going to take a higher level of care to clean that in that kind of environment. Or are they just regular vegetative bacterial cells that don't form spores? Then they're a little easier to control their growth. Our primary targets of control when we're thinking of cleaning is we want to look at organisms that can either cause infection, so make us sick, or cause food spoilage, and then that spoiled food makes us sick. So vegetative bacterial cells and endospores can do this, fungal hyphae and spores and yeast, protozoa and trophozoites and cysts, helminths, either the actual helminth or eggs, viruses, and prions. So these are our targets. These are our big ones. We know that there are many bacteria that don't cause harm that are actually helpful. We'd really not like to kill our good bacteria if we can help it. But at the same time, we do need a level of hygiene that allows us to be safe from pathogens. The degree that we have to control the growth, or the degree that we have to exercise extreme measures to prevent growth, depends on the microbe and their resistance to destruction. So on a scale of how resistant microbes are, enveloped viruses are pretty easy to destroy. That envelope is a phospholipid bilayer. We have lots of ways we can break that bilayer down and get to the protein structure. Viruses have no metabolism, so they can't put energy into maintaining their shape and their structure, and without that, they're no longer a threat. So envelope viruses, fairly easy to kill to destroy, because once we get through that envelope, if we break that envelope, we're also probably going to break that protein coat as well. Most gram-positive bacteria are fairly easy to control. And a lot of times people think this is counterintuitive because they have that big, thick layer, the multiple layers of peptidoglycan. But as we saw when we looked at binary fission at bacterial growth, in order for bacteria to grow, to increase in numbers, they have to be very metabolically active, constantly adding new peptidoglycan to that cell wall when they divide, building new wall to expand, and then to build the septum, the division, to create two new bacteria. So they're metabolically active, and metabolism gives us targets. Building that that thick peptidoglycan wall, if we can interfere or interrupt the building of that wall, that's the end of the bacteria. So that's a pretty easy target for us. It's also fairly easy because we don't have peptidoglycan. So if we target the destruction of that, our cells don't care, right? We don't have peptidoglycan. Non-enveloped viruses tend to be a little more difficult to kill. That layer of our membrane that the enveloped viruses take with them, with those spikes through it, when we break that, it's kind of holding on to that protein coat and to those spikes, and it starts to break that when it breaks down. Non-enveloped viruses, that external protein coat, the capsomere, is going to be the outermost structure, and so it's going to be a little more sturdy, more difficult to break down. Fungi and fungal spores. These are a little more difficult to destroy. Remember, our fungal spores are not are vegetative spores. They're not endospores like bacteria. They're not a protective structure. These are a reproductive structure. So they're metabolically active. They're a little more difficult to destroy than viruses or gram-positives. A little more difficult to target because they're eukaryotes, but different enough from us that they give us some targets. But they're fairly easy to destroy, easier than the gram-negative bacteria, and that's because of that outer lipopolysaccharide layer. So remember, they have a plasma membrane, just like fungal spores, just like gram-positives, just like protozoans. All living organisms have a plasma membrane or cell membrane. Those terms are interchangeable. They're synonyms. So plasma membrane occurs around all cells. Cell wall, peptidoglycan cell wall, is only found on bacteria. Fungus have a chitin cell wall, and plants have cellulose, so different structures. But our gram-negative bacteria, as well as plasma membrane, which is part of the cell, then that peptidoglycan layer, which is outside the plasma membrane, and then we have that third layer, that lipopolysaccharide lipopolysaccharide, it's lipids, it's fat, and polysaccharides, sugars. So this is difficult for a lot of things to get through, right? Those lipopolysaccharides, the lipids, will prevent, because of their polarity, they'll prevent a lot of molecules from getting in. They just give a whole extra layer of protection. So gram negatives are more difficult than gram positives. Protozoan cysts, in this case, we have a eukaryotic cell. The encysted form, this is their dormant stage. And so those dormant stages are always a little more difficult because it's easier to target metabolism. If something's actively carrying out life processes, it's easier to destroy it than when it's dormant. Staphylococcus and Pseudomonas are two exceptions to our order of most gram positive, most gram negative. Staphylococcus and Pseudomonas are more difficult. They're pretty high up on our scale. So if you look at, everybody has disinfecting products in their house, lots and lots of them. For a lot of them, you'll see kills gram positive, gram negative, and Pseudomonas. So they'll specifically list Pseudomonids as one of the things that they kill above just most bacteria and viruses. They'll specifically state that because it is more difficult to kill. And Pseudomonas is ubiquitous bacteria. It's present everywhere: freshwater, saltwater, lakes, your tap water. And it's usually typically harmless, but it is an opportunistic pathogen. And so because it's present everywhere, immunocompromised patients are particularly susceptible, especially burn patients. And then Staphylococcus, some strains like Staphylococcus aureus are of particular concern because they've become antibiotic. Some strains of them are antibiotic resistant. Mycobacterium, remember these are our bacteria that, while they should look like gram positives, they don't stain like gram positives because they have mycolic acid around the outside. That's a really protective coating. Remember, our mycolic acid is a waxy coating that makes it really hard to get at the bacteria with any sort of disinfectant or antiseptic. And then bacterial endospores, of course, are a dormant stage that can last forever. We've protected our DNA. We've protected just what we need, and we can survive in that dormant stage through a whole lot of assaults through disinfectants and antiseptics and physical and chemical methods trying to destroy those spores. And then our most resistant, as we learned, are prions of all weird things, an unprotected protein. This is just a protein that triggers misfolding of other proteins. It is a chemical compound. It's a protein. It's a chain of amino acids. It's not metabolically active, has no metabolism. It's not a living organism. It's not even as complex as a virus. It's just proteins. But they're very, very, very resistant to destruction. So when we're in the lab, what type of control methods we have present and we use is often dictated by or determined by the level of hazard based on what we commonly work with. So the CDC and the National Institutes of Health have a classification system that gives 4 different levels called biological safety levels. It's determined by an agent's infectivity. So what are we working with in our lab? Let's list everything we're working in and determine how infective is it? What's the ease of transmission? What's the severity of any disease it can cause? And what type of work is being done with the agent? So the designation of any lab is based on the highest biosafety level of organisms used. So you may mostly work with very innocuous organisms, but if you work with one that is a little bit more of a danger for any of these criteria, then you will be at that higher biosafety level. So our biosafety level classifications as we go up depends on the risk of the microbes. How much of a risk do they present to human health. And so a BSL-1 deals with non-pathogenic strains of E. coli, for example. So just organisms that are not known to cause any disease, that pose a minimal risk. So very just common safety practices, common sense practices. Biosafety level 2, we usually have indigenous bacteria, those that are part of our normal flora. And some of these are associated with disease of varying severity. Some could be very common and have mild diseases associated with them, and others a little more risky, but for the most part, they pose a very moderate risk to anyone working with them. Staphylococcus aureus is an example of this. And Staph aureus in the lab, most cultures, if you are not dealing with the methicillin-resistant variety, can be considered relatively safe. Microbes that are indigenous or exotic. So we have exotic, meaning they're introduced from somewhere. They're not normally found, either in the area or with the organisms being studied or associated with humans, with human normal flora. And have the ability to cause serious or potentially lethal disease, particularly through respiratory transmission. Because that one, we can work in hoods, we can have respirators, but if they leave that area and are transferred through the air, through duct systems, through air systems, air conditioning and heating units, just through the air, they're very easy to spread and contaminate. So Mycobacterium, the organism that causes tuberculosis, is an example of one of those. And then BSL-4, these are extremely dangerous. They pose a very high risk of aerosol transmission, of infection, and they are frequently fatal. So there are very few labs. They have very strict protocols when working with these viruses. For example, they'd be working with Ebola. They'd be working with SARS-CoV-2. So very strict requirements, and there are very few labs that can meet those requirements. When we're meeting in person in labs at Trident Tech, we are BSL-2. We do work with some organisms that are known to cause disease, but for the most part, with normal, healthy adults, our immune systems are more than capable of addressing any possible threat. All right, so some of the terminology. These are the words you're probably all familiar with, so really pay attention. Flashcards, quiz yourself, have your family quiz you, so that you can work through these vocabulary terms to differentiate. They are similar, but have specific meanings. And an incorrect meaning, even if it's off by a little bit, could mean that you are giving the definition of one of the other words. It makes your definition wrong. So you must be very precise with these. So when we talk about sterilization, we're talking about the complete removal or killing of all the vegetative cells, the endospores, and viruses from the targeted item or environment. So sterilization is our most stringent. We are removing everything, including endospores. A sterilant is a chemical that can be used to achieve sterilization. Aseptic technique involves both protocols of how to handle organisms, how to transfer organisms, as well as how to protect ourselves from being contaminated or from spreading this. So aseptic technique, protocols that collectively maintain sterility. So protocols, we've got rules, regulations, and behaviors and practices, including chemical or physical control methods we need to use. Asepsis is a sterile state resulting from the proper use of those protocols. Sepsis, as opposed to asepsis, A is without. Sepsis is a systemic inflammatory response to an infection, resulting in high fever, increased heart and respiratory rates, shock, and possibly death. So toxic shock syndrome is actually sepsis. So it's an inflammatory response. It's our own body trying to fight off an infection. So If you think of sepsis as being the bad one, A without sepsis. That's what our goal is in sterilization. So sterile fields are areas that are designated to be kept free of all vegetative microbes, endospores, and viruses. And having a sterile field is, of course, important in all sorts of medical practices and in surgery. It's not always necessary or practical to sterilize everything. To eliminate organisms from vegetative cells all the way through endospores, sometimes all we need to do is disinfection, which is going to inactivate most microbes on surfaces of fomites, of inanimate objects, tabletops, door handles, glasses. And all we need to do is inactivate most of the microbes using antimicrobial chemicals or heat. So disinfectants are antimicrobial chemicals that are applied to an inanimate object to achieve disinfection. Disinfectants are not used on living tissue or in living organisms. They can harm living tissue. Disinfectants are for use on surfaces. Antiseptics, again, we see sepsis, anti against it, are antimicrobial chemicals that can be used safely on living tissue. This is on the surface of living tissue. Again, not internally. So this on the surfaces, disinfectants are what we would use to clean tabletops. Things like Lysol, right? Disinfectant. It's on surfaces of inanimate objects. Antiseptics are things that we use on our tissue in order to reduce the number of microbes, to inactivate most of them. If we eliminate the large numbers, the majority of them, we have probably brought the number down to a safe level that our own bodies, our own immune systems can prevent from causing infection. Degerming. This word specifically refers to protocols or practices that significantly reduce microbial numbers by using mild chemicals like soap and then the mechanical action of gentle scrubbing. And that can be skin or tissue. Degerming, we can degerm surfaces. Sanitation is what we call that if we are degerming an inanimate surface. Right? So sanitization, if something has been sanitized, it means we've reduced the microbial load to a point that it's deemed safe for public health. So we haven't gotten rid of all of the microbes. We haven't gotten rid of the entire microbial load. We've reduced it again to the point that it is safe for most people, as long as you're not immunocompromised. It's a safe level. So we've got this table giving us, you spend some time going over some common agents. So disinfectants, some common agents, chlorine, bleaches, phenols like Lysol, sanitization to reduce that load, commercial dishwashing, right? So when we put our silverware in the dishwasher at a restaurant, that's sanitization. Clean public restrooms, which sanitize. So a lot of detergents and industrial strength compounds like quaternary ammonium compounds. We'll look at these a little more closely ahead. Sterilization, when we want to completely eliminate vegetative cells, endospores, and viruses from inanimate objects. So, for example, if we're preparing items for surgery, then we are going to use high pressure, high temperature autoclaves, or chemicals, or some types of chemicals, just a few, and then radiation. Those are all for use on fomites, on inanimate objects, surfaces, not on human tissue or inside of humans. For use on living tissue, we use antisepsis and antiseptic products like rubbing alcohol, hydrogen peroxide, or iodine. We clean skin that's broken due to injury or clean skin before surgery, using this to reduce the microbial load. And then degerming. This is our hand washing, our soap or use of alcohol swabs to remove microbes. One of the most effective ways to just keep people safe. All right. We need to differentiate when we're talking about control of growth between cidal and static. Bactericidal and bacteriostatic are two different ways that we can control growth. Right? So control of growth doesn't always mean we need to kill our microbes. Microbicidal or bactericidal, that cidal means killing. Right? So microbicidal agents are those that kill or destroy microbes. If you see that side, bactericide, kill bacteria. Fungicides kill fungus. Viricide kills viruses. And sporicide, this is a sterilant because it can kill endospores. Microbistatic or bacteriostatic agents just inhibit their growth. Doesn't kill them, prevents them from undergoing binary fission. So it inhibits the growth. They're temporarily prevented from multiplying, but they are not killed outright. Bacteriostatic prevents binary fission in bacteria, prevents their population from growing, but does not kill them. Fungistatic prevents the fungus from growing. A lot of the materials that are used to control microbes, either antiseptics on the body or in the body, and antibiotic drugs or chemotherapeutic agents, are microbistatic. And the reason for this is that microbicides, a lot of the chemical compounds that can outright kill microbes, are highly toxic, not only to the microbes, but to us. So a lot of the materials we use on the human body and in the human body are microbistatic. They prevent the multiplication of those bacteria, keeps their numbers from growing, and what that does is that buys our immune system time to take care of the infection itself. So how can we tell if we have a dead cell or if we have a static cell, a not dividing cell? Well, under the microscope, can't see a whole lot of activity in our bacteria, can we? So what are we going to look for? If we're looking for microbial death, the way we define that is that it's a permanent loss of reproductive capability, even under optimum growth conditions. So microbistatic or bacteriostatic agents will prevent bacteria from replicating. So if I put that, if I culture them on a plate that has a bacteriostatic agent in it, I won't see any colonies form. Do I know that it's dead or is it alive? I don't know. I would transfer that to a plate or a medium and provide it with its optimal growth conditions. And if it starts to multiply and forms a colony, then I know it was only static. But if it doesn't, then, it has a permanent loss of reproductive capability. So if I look at my microbial death curve, remember this is the end side, the right side of the growth curve that we looked at. And what we see is that microbial death can be logarithmic. And so just like the growth was logarithmic on one side, we are going to reduce the population decimal reduction time. When I plot this, I get the decimal reduction time, which is the time it takes to kill 90% of a population. So 90%. So I'm reducing this by 90% every time. That means only 10% left, and then 10%, and then 10% of that, and 10% of that. So 10%, then only 1/100th, then only 1/1000th of the population is left. So I plot that logarithmically to make it go from this drastic steep curve to something that I really can't see a change. to here where I'm able to count it. That's why we plot things on log paper, so that when we have that steep curve like that, oh, okay, I can figure out after time what my change in numbers are. Factors that affect the death rate. So how fast are things going to die? How fast is this curve going to decline? The effectiveness of any particular control agent or microbicidal agent is governed by several factors. This is really important. You can see a lot of examples of poor microbial control, growth control going on right now, as people spray with disinfectant and then wipe the surface immediately, or spray with disinfectant and then wipe multiple surfaces using the same cloth over and over. So some of the things that govern an control agent's effectiveness are the length of exposure. Read any disinfectant. The directions will tell you to spray and leave on for 30 seconds, a minute, 5 minutes, depending on what it is that it is designed to do. It needs time to do its job. The size of the population. The more microbes I have, the longer time it takes to sterilize or to kill all of those. So these are related. The larger the population, the longer the length of exposure, as well as just the length of exposure needed for the chemical action to take place. The nature of the microbes in the population, we looked at that scale of the least resistant to most resistant, depending what I have in that population or in the community of microbes that are present. I might need more time. I might need more of the substance, a higher concentration. So I need to know, what do I expect to be in that group of microorganisms I'm trying to kill? So then the concentration or the intensity of the exposure, the presence of solvents or organic matter or inhibitors. So solvents, organic matters, and inhibitors can all break down that control agent. Organic matter takes a lot longer. A lot of these compounds will act just on organic matter. So vomit, feces, urine, all of these things, if they are present, slow down the rate and the effectiveness, slow down the effectiveness of the organism in reaching, or of the compound in reaching the organisms. So how effective it is going to depend on length, size of the population, the number of microbes, the mode of action. So if I add the agent at this point, if it's microbistatic, it'll happen sooner. Sometimes a microbicidal agent is not initially microbicidal. It happens over long exposure. And so if I reduce the time of exposure, it's only microbistatic. And once I've removed that item, the microbes will continue to replicate. So I have to know how long before it's effective as a microbicidal agent. And then the nature, if I have spores, they are a lot more difficult to kill than vegetative cells, so it takes longer. All right, so let's look at some of the specific methods that we can use to control microorganisms. Some physical methods. There is a table summarizing this on pages 576 to 577. So in 13.2, I know the online textbook doesn't have page numbers, so just kind of flip around until you get to section 13.2. So our physical methods that we're going to look at include using heat in a number of different ways, refrigeration, so temperature, and then some other physical methods like high pressure, desiccation, lyophilization, osmotic pressure that we've talked about, and radiation. So as we go through these slides, the modes of action. How does this disrupt the bacteria? That's going to be written in red. If the method is able to sterilize, meaning it is able to eliminate endospores as well, it'll be noted. So you want to pay attention. Those are important things to know. So look at heat. both moist and dry heat, cold temperatures, high pressure, desiccation, lyophilization, osmotic pressure radiation, and filtration. So with heat, our moist heat, like boiling, requires lower temperatures and shorter exposure time to destroy microbes than does dry heat. So moderate to high temperatures takes longer exposure time. So my dry heat's a little less effective than my moist heat as far as how effective, how efficient, how fast it's going to happen. The way both of these work is they denature proteins. Remember, my proteins are polypeptides of amino acids, and they are held together in their very specific shape to do their job by hydrogen bonds, and adding heat breaks those hydrogen bonds and denatures my protein. It loses its shape. It can no longer do its job. Metabolism, the sum total of all the chemical reactions in a cell, all of metabolism, happens because of enzymes speeding these chemical reactions up to the rate needed to sustain life. I denature my proteins and I stop metabolism. I can measure the effectiveness of different heat-related control mechanisms by looking at things like the thermal death time. I can compare the thermal death time between two different methods or ways of heating, between moist heat, dry heat, boiling, steaming, things like that, and compare their thermal death time. The thermal death time is the shortest length of time that I need to kill all the test microbes at a specific temperature. So the thermal death time, the time it takes to kill all those microbes at a given temperature. So the temperature is specified. How long is it going to take to kill these microbes? I could also look at it from the other side, the thermal death point, where I'm given a time. So in 10 minutes, what's the lowest temperature required to kill all the microbes in a sample in 10 minutes? So these are looking at efficiency. If I am in a commercial use or a hospital setting and I want to kill everything, I want to do it either at the lowest temperature possible. So what's the temperature I need to kill all the organisms in 10 minutes? Or I want to do it as quickly as possible. So what's the temperature I need to do it in the shortest length of time? So you can use either one of those to compare methods. Oh, there we go. I can look at moist heat methods by comparing these methods. Boiling water. So if I boil water at 100 degrees for 30 minutes, it will destroy non-spore forming pathogens. This is disinfection. Pasteurization, I'm going to apply heat to liquids so that I can kill any potential agents of infection and spoilage, but I don't want to do it so high, so hot, or for so long that I also destroy the food, either the flavor, the value, the texture. So in this case, I'm probably going to end up leaving some microbes behind. That's why even though your milk has been pasteurized, it won't stay indefinitely in the refrigerator. There are still some organisms left behind there. So this is disinfection. It kills non-spore forming pathogens and it lowers the overall microbe count. So why wouldn't I just want to go ahead and kill everything by raising the temperature some more or doing it for longer? If I start to break down either the texture, maybe of milk, it's hard if I boil that. If I did that first, milk would not work as well in cooking. I would lose the flavor. So we pasteurize instead. We bring it to a safe level and package it in amounts that we should be able to use up before spoilage begins, before those microbes begin to grow. Ultra high temperature pasteurization. This is relatively new. That's our boxed milk on the shelf that will last a long time. I can heat the milk at a really high temperature, 138 degrees, so quite a bit higher than 72, for 2 or more seconds, really quick, and then seal it in an airtight container. So ultra high temperature pasteurization gives me some benefits that allow this milk to remain fresh longer. Some other moist heat methods include autoclaves, steam under pressure. So autoclaves are commonly used for sterilization in laboratory and clinical settings. The air, we load everything into this. It looks like a big giant dishwasher. Load everything in, displace the air with increasing amounts of steam, and the pressure is raised. So we have high pressure and temperatures. We exceed temperatures of 100 degrees Celsius, which is boiling, and achieve complete sterilization. Dry heat, as opposed to moist heat, where we're using steam or boiling, we need higher temperatures than moist heat. I can achieve sterilization. So here I could achieve sterilization with my autoclave. Here, dry heat, I can achieve sterilization because I'm going to incinerate. I'm going to physically destroy my microbe. So incinerators either use a flame or electric heat with a coil. So in our labs, we used to just use open flamed Bunsen burners for incineration. But here we have incinerators that use an electrical heating coil, much safer, not having an open flame around. And the mode of action is that we actually ignite and we reduce the microbe to ashes and gas. So we completely destroy them. That's why it's sterilized. This is limited to metals and heat-resistant glass. Obviously, paper and human tissue don't do so well if we put them in an open flame or in high heat in an electric heating coil, because they would also ignite and be reduced to ash. So we're limited in how we can use dry heat. We do use dry heat in ovens. So that's, our oven, when we put things in, that is dry heat. And we can lower the temperature and just keep things in for a long period of time to achieve sterilization. Cold temperatures. Cold temperatures inhibit growth. So if you remember in our control of growth, we looked at temperature as a way to control growth, where high heat can destroy, but low temperatures inhibit growth. So refrigeration is between 0 and 7 degrees Celsius. Freezing is below 0, or we could use ultra low temperature freezing. And we use these to preserve food, preserve media, and preserve stock cultures in the lab. So if I want to hold on to a sample for a while, I can refrigerate or freeze it, and subsequently when the temperature increases, those cells will still be viable. So remember, we have our optimal growth temperature. Once we get up here, enzymes denature, bonds on those proteins, those hydrogen bonds break down. I can physically incinerate my cell at high enough temperatures. So those that exceed the maximum growth temperature are microbicidal because of those, and all these effects are irreversible. But on this side, the cooler side, this is just going to inhibit growth by slowing down molecular motion, chemical reactions, metabolism slows. Will slow till we have no growth, no replication, but the cell is not destroyed. Those effects are reversible by increasing temperature. Can use high pressure. It's called Pascalization, and it can kill microbes in food while maintaining the quality of those foods. Endospores can survive this process, so this is not sterilization. And these pressure containers, a lot of home canners will know about increasing the pressure by boiling and then sealing that in under high pressure as a way to control growth. Desiccation is dehydration. I gradually remove all of the water from cells. Without that water, I've inhibited metabolic activity, because metabolism, the sum total of all the chemical reactions in a cell, those chemical reactions take place in an aqueous solution in the cytoplasm of the cell. So this is great. This is beef jerky, dehydration, food dehydrators. They can inhibit that growth, but it's not really effective for a control mechanism for microbes because many of them, once water is reintroduced, they can grow again. It's actually how endospores germinate. Is water is reintroduced along with nutrients in a proper environment, but they do need water. Lyophilization is freeze drying, and we can preserve foods and microbial cultures that way. It's microbistatic. It only prevents replication, so growth of the population. It doesn't kill it. This is actually when we order microbes to use in the lab, all the bacteria that we use in the lab, order them online and they come freeze-dried. And then we just have to give them water, the right temperature, and some food, and they begin to grow again. Osmotic pressure. So we've spent a lot of time on osmosis, and you are doing an activity this week on osmosis. Remember, as you do that activity, it's all osmosis. And osmosis is a special case of diffusion where we're only looking at which direction water is going to move across a membrane by comparing what the solute concentration on either side of the membrane is. So we can use osmotic pressure by creating a hypertonic solution or environment outside of the bacterial cells, that hypertonic solution will draw water out of the cells and no multiplication. This causes plasmolysis. Plasmolysis is when the plasma membrane pulls away from that cell wall. So normally the cytoplasm, the cell is filled with cytoplasm, our solution inside, our aqueous solution inside the cell, the water with all the dissolved resources the bacteria needs, and that presses the membrane up against the peptidoglycan cell wall layers. As the water is removed from that bacterial cell, the peptidoglycan is a physical structure. It's not metabolically active itself. It's just a physical structure giving the bacteria shape. So it will retain its shape, but inside, as that water leaves, the plasma membrane pulls away from that wall and shrivels in. That's plasmolysis. We won't see any change under the microscope, or very slight, because that cell wall will retain its shape. But inside the cell, just like in plants, in plasmolysis, that membrane pulls away from the cell wall. This is microbistatic. Again, return water balance, remove the salt, and these organisms can become metabolically active again. So radiation is another way we can control bacterial growth. Radiation is energy that's emitted from atomic activities. So it's electrons bouncing off of different elements. So those electrons are dispersed at high velocity through matter or space. We have two different types of radiation. We have ionizing radiation. Ionizing radiation has deep penetrating power. It can cause electrons to leave their orbit, enter into a cell, deeply into a cell. That deep penetrating power can penetrate in through the cell, and it can break DNA. And so those ionizing radiation, X-rays are an example, gamma rays, they can pass all the way through the cell to its DNA and start breaking those double strands of DNA. That's how it manages to kill the cell. No DNA, no directions, no ability to replicate the DNA, no ability to divide. So ionizing radiation can achieve sterilization. We use gamma rays or X-rays or high-energy electron beams, and we can use this to sterilize medical supplies and food products. Obviously, we don't use this on living tissue. Damaging DNA, doesn't care if you're a bacteria or a person, it can still damage the DNA. So we do use ionizing radiation on food to preserve it. Because if the DNA in the food cell is broken, or in the food that we're about to eat is broken, we don't care, it's just broken DNA. We're going to eat it and digest it and break it down anyway. So we can use this on our food sources to sterilize or to preserve food. So we can preserve our food with that by removing bacteria or organisms that cause spoilage. And we use gamma radiation. And they are labeled so that people know this has been exposed to radiation. The radiation is gone when we get it, but it has been exposed. So that is labeled. We also have non-ionizing radiation. Non-ionizing radiation doesn't penetrate very deep. It needs to be, whatever we're trying to sterilize or we're trying to disinfect has to be directly exposed to non-ionizing radiation. So we see the germicidal lamps, those UV lamps, the blue lights. We used to see them. They used to be in doctors' offices all the time. With COVID, now we have handheld UV lights that are being used to disinfect around rooms and places and materials that come into contact, potentially exposed by an infected person, as a way to interfere. Again, it interferes with the DNA in a very specific way. It creates thymine dimers. So anywhere in DNA where we have two thymines next to each other, it will cause them to bond to each other on the same strand rather than forming their complementary bond with adenine on the other strand. So it will disrupt the DNA, and by doing that, it disrupts the ability of the DNA to replicate or to undergo transcription. So we can use that non-iodizing radiation, but it does have to be directly exposed. So if nooks and crannies or hidden areas, it wouldn't work. It's got to be that light has to be directly exposed, or the surface has to be directly exposed to that UV radiation. Another way that we control growth is through filtration. Filtration is the mechanical removal of microbes. So this is a physical process. We physically remove the microbes using a filter. So we can pass either gas or liquid through a filter, depending on what the pore size of the filters are. We have different degrees. We can select the pore size. So we put a filter here, and we would pass a liquid through here. We'd pour the liquid in, a pump that's pulling out oxygen that just makes it flow faster. And as it flows, the liquid flows through, we select this filter paper based on the pore size. If we're looking at something larger, we can have bigger pores. If we want to filter out smaller size things like viruses, we would have smaller pores that would be caught on this filter. So depending on this, we can achieve sterilization or we can just disinfect. And we use this with liquids that are sensitive to heat. So we use this with air systems to filter particles out. And depending on the size of the pores in our filter, we have different levels of filtration. So now everybody's familiar with HEPA air filters. 99.5% of the particles of a certain size are filtered out. And so this is our most effective that's typically normally used. So we can filter liquids and gases through those. And this, again, it's physical or mechanical, and we're just removing the microbes from either that liquid or gas. We can also use filtration to concentrate microbes and grow them if we want to know how many. So we talked about enumerating bacteria. Filtration is a way we can do that. For example, with seawater, I want to know how many bacteria there are in a liter. I would filter a liter of water through this, and then I would put this filter on a plate or in an environment that the bacteria would grow. So I could just put it on the surface of an agar plate and incubate it and see how many bacteria I have. So filtration is another method I can use to count my bacterial numbers. All right, so those were all physical methods of control. So look at some chemical methods. Here we have a table on 593 to 594. Again, you have no page numbers, so go to section 13.3 and scroll till you find it. And we will compare some different chemicals that are used to control microbial growth and look at if they are disinfectant, antiseptic, or sterilants. Sometimes we have things that we do use as antiseptics that can also be used as disinfectants, or we use them as disinfectants and they are safe for use on tissue, so they can be both. And then we'll learn a little bit about some of these chemical agents and when we would use them, like phenolics, metals, halogens, alcohols, surfactants, bisbiguanides, guanides, alkylating agents, peroxygens, supercritical fluids, and then chemical food preservatives and natural food preservatives. So these are our groups, same as before. Modes of action are in red, and if it can achieve sterilization, that will be noted. Phenolics. These are going to be compounds that disrupt cell membranes and denature proteins. So they will interfere with the cell membrane. They'll get in between those phospholipids. They'll denature the proteins by destroying the plasma membrane. They allow things that shouldn't be in the cell to enter the cell. Right, so ions and chemicals are able to enter the cell that would normally be kept out by that membrane, and all the stuff inside the cell, the resources it needs, would leak out. Not good for the cell. This has low to intermediate level of bactericidal, fungicidal, and virucidal abilities. It is not sporicidal. Phenolics will not kill endospores. So Lysol is an example. If you read the label on your Lysol that you have around the house, triclosan is an antibacterial additive in a lot of soaps that is under close scrutiny. Probably don't want it in your soaps. So remember back to, I think, chapter 3, who first used phenol as a surgical germicide? So to say carbolic acid is a phenol, commonly used phenol. So do you remember who that was? If you're saying Lister, you're correct. So triclosan, it's a common ingredient in antibacterial soaps, but it has a really high environmental and health risks, including being a possible mutagen and carcinogen. So triclosan, once it gets into the environment, it disrupts a whole bunch of metabolic processes in aquatic organisms. So it's come under a lot of scrutiny. It probably should be banned. It's banned in Europe. It actually is not any more beneficial having antibacterial soaps. Turns out they don't do any better at all than just washing your hands with conventional soaps, soap water, 20 seconds, sing happy birthday twice. And lo and behold, we have the exact same results for cleanliness and hygiene, only without introducing harmful chemicals to your body or to the environment. Heavy metals. So a number of heavy metals, things like silver, mercury, copper, kill vegetative bacterial cells, fungus, algae, protozoans. I think anti-fouling paints on boats are anti-fouling because they contain mercury. They contain heavy metals. Some are moving more toward copper as a more environmentally safe option, but it's doing the same thing. It's killing living organisms. They are general biocides. They don't really care what kind of cell you are. And the way they work is these heavy metals will bind to and inhibit the activity of proteins. So they will bind there into the enzymes and prevent them from doing their job. And remember, no enzymes, no metabolism, and that's the end of the organism. They're oligodynamic. Oligo is many, so they have oligodynamic action. They're very effective in small quantities. Just a little bit can affect many different proteins, many cells. They're very active. Mercury, silver, copper, nickel, zinc. These are all heavy metal examples. They denature proteins, so cell function stops, cell metabolism can stop. There's more and more use now looking at incorporating these in into medical uses. So medical structures like hospital beds, we'll talk about that in a minute, but copper fixtures would kill microbes that otherwise would accumulate on this foamite. Having small amounts of silver or other metals in silverware and frequently touched surfaces can reduce their microbial load. Bad part of this is that heavy metals can accumulate in our body, and by bioaccumulating, they interfere with our own metabolism. This is an irreversible condition, argyria. Argentina is named after the Latin word for silver. Argentine, argyria, is a bioaccumulation of silver in the body. So they have good control of microbes. This is local interest here. Copper is linked to a reduction of bacteria in hospitals. And you can go in and click on this link on the TED Talk, where they've started to incorporate copper compounds on the metal components on the handrails on hospital beds, and it's reduced infection at a very high rate. So that is a very interesting TED Talk you might want to go take a look at. It's going on at MUSC, some clinical studies. So halogen compounds, so iodine, chlorine, bromine, fluorine, these are all halogens. So iodine is I2, is iodophores, betadine. Everybody's familiar with betadine solutions used for first aid. And this oxidizes cellular components. So remember, oxidation is loss of electrons. Those electrons will be drawn from bacterial, from membrane components, from cell wall components, and it oxidizes them. They're no longer doing so very well. So this is used as a topical antiseptic. They are fairly mild for use on human tissue or on living tissue. So iodophores are generally used on tissue. Chlorine is a halogen, as hypochlorites, which are used in chlorine bleaches. These are strong oxidizers. So these are pretty damaging to human tissue. So can use it in very small quantities in drinking water. Will disinfect very well because it is such a strong oxidizing agent. It's used in sewage and used to clean inanimate objects. Fluorine, like fluorides, are incorporated in tooth enamel. So they accumulate in plaque-forming bacteria and interfere with their metabolism, which is why we add fluoride to drinking water. Also add that to wastewater. Alcohols. So alcohols are interesting because for most of these chemical compounds, the higher concentration, the more effective they are. With alcohol, they're most effective at 70%, around 70% concentration. So at less than 100, so more than 50, but less than 100% concentration is where they are most effective. Ethyl alcohol, isopropyl alcohol, rubbing alcohol are the most commonly used forms. We can use them as antiseptics. We can also use these as disinfectants, so on surfaces or on tissue. They dissolve membrane lipids and denature proteins in vegetative bacterial cells and fungus. Notice, not on endospores. So these are going to dissolve. You notice it in the staining process when we decolorize. We decolorize using alcohol, and that breaks down the membrane lipids. That's where all the colorization from our primary stain is on our gram negatives. It's all caught up in that lipopolysaccharide outer membrane, whereas it goes right into the peptidoglycan and the gram positives. So then we use this to dissolve that membrane, and that's what decolorizes our gram negatives in the gram staining process. Surfactants. Soaps are surfactants. They are surface acting agents. they mechanically lift. You see this in a lot of commercials on soaps. Lifts and washes away grease, stains, microbes. So soaps mechanically lift and remove soil and grease that contain microbes. So any of the dirt on our hand, they lift away just our normal oils that are in our skin, lifting that away, flush it down the drain. So soaps are made of the salts of fatty acids. So they emulsify lipids and fats. That's how they lift them off. They get in between, they break them down, and then they interact with the water, and off we go, rinsed down the drain. Quaternary ammonia compounds, slightly different than soaps, but similar. So you see these two used together because they're both surfactants. These lower the tension of water and alter membrane permeability. So alternating membrane permeability means they get in between these phospholipids in our phospholipid bilayer, opening it up for ions and other chemical compounds to come in, and for the components of the cytoplasm, the resources the cell needs to survive, to go out. We bust this open. They're able to infiltrate that membrane, disrupt the membrane integrity, and lead to cell death. Bisbiguanides, these are chlorhexidine, alexidine. These are skin degerminating agents that are used as preoperative scrubs to clean skin prior to surgery. They're also used to clean burns and are also used as an oral rinse. These are broad spectrum and have lots of different microbicidal properties. They disrupt the cell membrane and cytoplasmic congealing, so cause cytoplasmic congealing. So the membrane opens up, the cytoplasm gels or solidifies, and that's the end of the cell. Alkylating agents. This is formaldehyde. This is what we preserve specimens in when we're going to dissect them in the lab. I actually use formalin now. It's a watered down version of it. But formaldehyde is a disinfectant and a preservative, highly toxic, and so don't tend to use it a whole lot on human tissue or around people. Glutaraldehyde, it's used as a sterilant for heat-sensitive instruments. And how it acts is it replaces hydrogen with an alkyl group, and that inactivates enzymes and nucleic acids. So our enzymes are carrying out metabolism. Our nucleic acids are the directions for all of that metabolism. So by interfering with that, we are bactericidal, but also highly toxic. So not the best choice. Another alkylating agent, and one that is able to achieve sterilization, is ethylene oxide. Ethylene oxide is a gas, and it's used to sterilize and disinfect plastics, prepackaged foods, and a lot of medical devices. You put the product into a gas into this chamber, and we pump in the ethylene oxide, and it sterilizes. Peroxygens, we have another oxidizing agent. These produce highly reactive free radicals. So remember, we had to have our catalase, our enzymes to deal with those toxic oxygen byproducts. We talked about that a little bit with gas requirements of organisms. So we can use those free radicals, we can use those toxic oxygen compounds to kill especially anaerobes. So hydrogen peroxide. Usually over-the-counter hydrogen peroxide is 3% mixture. We can use it up to 25 as an antiseptic or disinfectant. And we use it to kill organisms. Aerobes have enzymes that are able to break down hydrogen peroxide into harmless water and oxygen. Anaerobes don't have that, and that's why oxygen is toxic to them. They have no way to break down hydrogen peroxide. So when we see bubbling, that means we have put our hydrogen peroxide onto anaerobes that are able to break it down, or onto our own damaged tissues. And when we damage that tissue, cut open those cells, we release our own catalase that causes the hydrogen peroxide to bubble. If you don't have an enzyme that breaks it down, though, this is toxic, so it will kill anaerobic bacteria. In the gaseous form, it can be used as a sterilant for rooms or equipment. So supercritical fluids. Supercritical fluids are those that have properties that are somewhere in between a liquid and a gas. Carbon dioxide at high pressure, so 10 times atmospheric pressure, is in a physical state that it's not really a liquid, it's not really a gas, it's somewhere in between. It's a supercritical fluid. It's able to penetrate surfaces and sterilize. To kill microbes, it penetrates the cell, forms carbonic acid, and that lowers the cell's pH, and that's going to interrupt a whole bunch of metabolic processes by destroying enzymes and a whole lot of other components of the cell. If we use this in combination with peracetic acid, it can also kill endospores, so that's when we get the sterilization. It's non-toxic, non-flammable, and non-reactive, so it's a good option for treating foods and medical devices. We have some chemical preservatives in foods that inhibit growth and minimize spoilage. Many of these decrease pH, so they inhibit the function of enzymes, and that stops metabolism. So sorbic acid, benzoic acid, propionic acid are all things that are going to interfere with enzymes by decreasing pH and denaturing those enzymes. And we have some natural food preservatives that have antimicrobial compounds. These are actually produced by bacteria. So these bacteria, in order to outcompete other bacteria, produce these antimicrobial compounds, and we can use them as natural food preservatives. Niacin is produced by Lactococcus lactis, and natamycin is produced by Streptococcus natalensis. And these have antimicrobial compounds, so they can destroy other bacteria. All right, so I know that's a lot. It's a lot to work through. Not studying the tables that I pointed to you, but trying to recreate those tables is always a helpful way to work through this, and also learn one at a time. So don't try to learn, here are all the chemical methods of control. Look at one chemical method, control, an example, think about that, walk away, come back. So learning one at a time would only take 5 minutes, but then walk away and come back so that you can dissociate. Otherwise, they all become one big blur. Right? So this is its own separate item, and when I learn the next chemical method of control, it's its own separate item. All right, so that's my suggestion for a strategy. On to testing. Talk about the phenol coefficient and compare and contrast some different methods that we use to test. So the phenol coefficient historically has been used to compare any chemical agent of microbial control against the effectiveness of phenol. So how fast does phenol work compared to this? If we have a coefficient of 1, it means it's as effective. So this coefficient, it's a ratio. Right? So if 1 means it's a 1-to-1, it's a ratio. It works in this amount of time, phenol works in this amount of time to do the same work. If it is less than 1, then that means it's less effective than phenol. Right? If it's greater than 1, it's more effective than phenol. So don't use this a whole lot anymore, because at the time phenol was just sort of randomly chosen because that's what we had, that's what we used. Everything, the lister came up with phenol, or carbolic acid as a way to clean. So that's what things were typically measured against. Now we have a whole bunch of other ways to compare different agents to one another. The disk diffusion method, this is something that we typically do in lab. I'm not sure if you've gotten to this yet or if you will see this practice, but whatever the chemical agent is, you are trying to determine which one's best, which one's better. We soak little paper discs in that chemical. We take an agar disc and we lay down a lawn of bacteria. So you would just cover the entire plate with this same species of bacteria. Usually we'll use Pseudomonas and Staphylococcus. Remember our resistance, our difficulty to kill. One is a gram-positive, my Staphylococcus. One is a gram-negative, Pseudomonas. And then I will place these discs on that lawn of growth, and I will incubate this. And then I will compare the zones of inhibition. So a zone of inhibition, how far out did this prevent growth? Right? So I put this on before I had growth. So the growth that took place during incubation, where it was inhibited, I get this circle of no growth. And we call this disc diffusion because the chemical compound on this disc will diffuse out through the agar. So how far out it's going to diffuse can vary. So if I'm looking at antibiotics, I use a very specific type of media called Mueller-Hinton. But I can just compare, did it inhibit the growth or did it not? If I just use regular agar, regular nutrient agar. If there's no growth, okay, it's effective. It inhibited the growth. If there's growth around the disc, it grew right up to there, it did nothing to inhibit the growth. So I can, if I'm just looking to compare disinfectants, all I'm going to look at with the disc diffusion method is, did it inhibit the growth or not? Because different compounds diffuse at a different rate through the agar, the size of this is not comparable. I can't say this one's better than this one. It might have just diffused further through if I'm just looking at chemical compounds. If I use that Mueller-Hinton agar, I can look at the effectiveness of antibiotics or antimicrobials because I'm going to control the amount on the disc and I'm going to control the diffusion rate by using Mueller-Hinton. That makes the zone, how effective this is at inhibiting growth is related to how far this zone of inhibition extends. So I would measure that zone of inhibition. The larger zones would be correlated with greater inhibition. I have a use dilution test. And what a use dilution test is used for is to determine the effectiveness of a compound in preventing growth or inhibiting growth on inanimate surfaces. So what I'd do is I would soak these stainless steel cylinders in bacteria. So all of these have been soaked in bacteria, so I know there are bacteria on them. And then I would treat the carriers. I would treat these with a disinfectant. Then I would put them into growth media and grow to see, did any bacteria survive? If there's growth, if they could survive that disinfectant, then there would be growth once I put them into their optimal, gave them media and put them in their optimal conditions. If the compound, if the disinfectant was effective, then I would not get growth. This is not a quantitative, but a qualitative measure. Did I get growth or no growth? But I don't know, how much, how effective was it? Did it control it a lot? If I got growth, if I get any growth at all, it's growth. So it's a yes or no answer, not a degree of effectiveness. The in-use test. This I'm going to determine if I'm actively using disinfectants, a disinfecting compound. I'm actively using it. It can become contaminated. If I'm pouring it out of the same container, I can contaminate this over time. And so I want to know, is it contaminated? So what I would do would be to transfer, in this case, 1 ml of the disinfectant. Here's my disinfectant. It's been used. Is it contaminated? I would transfer it into this test tube that has a disinfectant inactivator. So this contains a compound that is going to deactivate my disinfectant. It will no longer be able to disinfect or inhibit the growth or kill the microbes. And then I'm going to put a sample of that onto a plate and I am going to incubate it and grow it. So think about this. Here's my disinfectant. If it's not contaminated, and I deactivate it, well, if it's not contaminated, it's still effective. There won't be any bacteria growing in here. So I deactivate it, and I should get no growth. Here, I deactivate the thing that's inhibiting the growth. So I've deactivated this. It's no longer inhibiting. This prevents it from inhibiting growth. Now my bacteria can grow. So that's how I can tell if it's been infected. All right, and that is it for this chapter. And this will get us through everything that's on our next exam. So go through these, take your time, organizing all of these different compounds and how they work. This will be the chapter you want to spend a lot of time on. And I will see you in the classroom.